This video is going to be all about DNA replication. So remember, the purpose of DNA replication is for a cell to make an identical copy of its DNA. That way, other cells also have DNA. So let's look at an overview of the process. Before I get started, I want to mention that this PowerPoint is not identical to the one that you see in Blackboard because this one is animated and is meant to help bring this abstract concept to life. I will post this PowerPoint afterwards, but printing it won't do you a lot of good because it's animated. So there's a lot of pictures stacked on top of each other. So if you do want to review this PowerPoint later, you're going to have to watch it in presentation mode. But the content in this video aligns with all the content in the main microbial genetics PowerPoint. Okay, let's get started. So as you learned in the previous video, there are a few important features of DNA. One is that it is a double-stranded helix and it is linear. And so that's what you're seeing here, the two strands of DNA. So remember that each strand has directionality. So in this example, the top strand runs 5' to 3'. And because these two strands are anti-parallel, we know that the second strand runs opposite, which means it runs 3' to 5'. Each strand that makes up the DNA molecule contains 4 different nitrogenous bases, which includes adenine, guanine, cytosine, and thymine. So I made up a DNA sequence, which is just the order of the bases. And remember that the two strands are also complementary. So that means that anywhere there's an adenine on one strand, you'll see a thymine on the opposite strand. Where you see guanine on the top strand, you'll see cytosine on the bottom strand. Okay, so complementary is another term for base pairing. Adenine always pairs with thymine in DNA, and guanine and cytosine always pair together in DNA. Okay, so you're going to have to remember that as we go through this process of DNA replication. So let's go ahead and look at big picture what's going on. So first of all, what happens is we're looking at the left-hand side of this slide. We start with a parent molecule. And right now, we're not worried about the DNA sequence. We're just looking at big picture what's happening. So you see the two anti-parallel strands that make up our starting DNA parent molecule. The first thing that's going to happen is those two strands are going to separate. That is possible because the bases that make up the two strands, they are joined, they connect to the opposite strand by hydrogen bonds. So let me actually go back for a second. So if you look at the adenine, the very first adenine on the top strand, remember I told you that it base pairs with the thymine on the bottom strand? There is a hydrogen bond, which is a weak bond, that's connecting that adenine on the top strand to the thymine on the bottom strand. So essentially, the two strands that make up the DNA molecule are connected to one another by weak hydrogen bonds. So those weak hydrogen bonds can easily be broken. Okay, and so that's what happens. Those hydrogen bonds are broken and the two parent strands are able to separate. Now that those two strands are separated, the cell can create two new strands of DNA that are anti-parallel and complementary to the two original parent strands. So if you look at the far right at the daughter molecules, you'll notice that each new strand that's made, which I showed in blue, is anti-parallel, which means it runs in opposite directions to the orange strand that it's with. And if I had put the bases in there, it would also be complementary. Okay, so we say that after one round of DNA replication, there are now two identical daughter molecules. There are now two copies of DNA. We say this process is semi-conservative because each of these daughter DNA molecules has one of the very original parent strand, but it also has one new strand, okay? So the fact that there's one original strand and one new strand that makes up each DNA molecule, we say it's a semi-conservative process. Because in biology, when something is conserved, like completely conserved, that means it is identical to what it was before. Well, in this case, each daughter DNA molecule has one brand new strand of DNA. So it's not the exact same parent molecule that we started with, right? It's got one of the old strands and one of the new strands. So we say that it is a semi-conservative process. Okay, so let's kind of look at this again, but I'm including some bases here. So we have our parent molecule. Again, the two strands are anti-parallel. The top strand runs 5' to 3'. The bottom strand runs 3' to 5'. You can see that the bases are anti-parallel. So as I mentioned on the last slide, the next thing that's going to happen is the hydrogen bonds that are keeping the two strands connected to one another are broken. So now those parent strands are separated. This allows the cell to use each of these parent strands as a template. Essentially, we can build a new strand of nucleotides that is opposite and that is anti-parallel and complementary to that top strand. The cell can also build another strand of nucleotides that is anti-parallel and complementary to the bottom parent strand. Okay, so that's what I'm going to show you next. So we're going to be focusing our attention at that top 5' to 3' strand. So we know that the cell needs to create another strand, right? Because DNA is a double-stranded molecule. So the cell is going to start building this new strand. And so you'll notice this new strand, which is in blue, it's running in the opposite direction. Notice it starts with 3', and each base that's being added one at a time is complementary to the base that is on the original parent strand. Okay, and so these nucleotides are just added one at a time until it gets to the end. And then you'll notice, once it's done, I'll show that the end of the strand is anti-parallel to that top parent strand. We can also call the parent strands the template strands because that's what we're using to create our new strands. Now, the same thing is going to happen for the bottom. And I apologize, I messed up my animations. So now that both strands are complete, the text at the bottom should say daughter molecule. That means we now have two complete pieces of DNA. Okay, so I apologize for that animation mistake. It should say daughter molecules. Now, if you compare these two daughter molecules, okay, look at the orange 5' to 3' strand and look at just the first few bases, A, G, C, T. We'll compare that to the bottom DNA molecule, 5' to 3' strand, A, G, C, T. If you compared the whole thing, you would see that those strands have the exact same sequence. The same thing goes for the bottom 3' to 5' strands. In both DNA molecules, they have the same directionality and the same sequence. So these two DNA molecules are identical. And that's important, right? When an E. coli cell is about to divide and create a new E. coli cell, it needs the DNA to be exactly the same, right? So we say that these two these two molecules of DNA are identical. All right, so now we're going to dive a little bit more into the details to talk about how that new strand is made for each daughter molecule. So again, we're not worried about the bases at this exact moment, but I'm showing you the directionality. Notice there's these little like tick marks that I put on there. DNA replication begins at specific sequences, okay? And those sequences are called the origins of replication. Now, this topic of the course is microbial genetics because we're talking about how prokaryotes and eukaryotes do some of these same processes. So please know that because the DNA is linear here, I am showing you the eukaryotic process. Okay, so my example of E. coli cells because we're talking about eukaryotic DNA, but know that the same process is happening with bacterial cells as well. But remember, their chromosome is circular, so it's just a lot harder to create images of this happening with a circular piece of DNA. But the same thing is happening. The big difference is, aside from the fact that bacterial DNA is circular, they only have one origin of replication because they have significantly less DNA than eukaryotes do, okay? So in this picture of eukaryotic DNA, there are multiple origins of replication because cells divide so quickly that we need DNA replication to happen as quickly and efficiently as possible, okay? So in order to do that in eukaryotes, there are multiple origins of replication on the DNA. Now, what happens next is when it's time for DNA replication to occur, the strand of DNA is going to separate only at the origins of replication, okay? And so see how it looks like there's little humps in the DNA, okay? So we call each of those openings a replication bubble, okay? Now, you can see where I put blue arrows in each of those bubbles. That is showing you that the DNA, the new DNA strand, is going to be made bidirectionally in each of those bubbles. Again, we're trying to maximize speed here. So once a bubble opens, the new strand is going to be made in two directions, on both the top strand and the bottom strand. So any part of the DNA that is still closed, it's still completely linear, that region of the DNA is not currently being copied. The DNA gets copied one kind of bubble at a time. Let me rephrase that. So each of these bubbles in the image are currently being copied. The new strands are being made, but only in the bubble region. And that's because once those little bubble regions have been copied or used as a template to create the new strand of DNA, the bubble expands. And so then the newly exposed regions of DNA are used as a template to create that new strand of DNA. And then, as you can imagine, once those bubbles have been completely used to make the new strand of DNA, they get even larger. This continues until the entire piece of DNA has been used as a template to create the new strands, and we have two daughter molecules of DNA. Okay, so again, just to be perfectly clear, the top two strands, the 5 to 3' and then the 3' to 5, that's one daughter DNA molecule. And then the bottom set is DNA molecule number 2. Again, this is a semi-conservative process. So that means each of those DNA daughter molecules has a parent strand. which are shown in orange, and they each have a new strand. Okay? All right. So just one more time to kind of give you a chance to try to wrap your head around this. At the origin of replication, when it's time for DNA replication to occur, little portions of the DNA separate, forming replication bubbles. DNA, the new DNA strand, is going to be made bidirectionally within that bubble. So the separated template strands are going to be used to create these new strands of DNA. Those bubbles get bigger and bigger as the regions are used to make the new strand of DNA until they separate. Okay? All right. So again, just to reiterate some terminology. So here's kind of, the big open replication bubbles. Okay, so replication bubble is a term you're going to hear. And so I've indicated all of the different replication bubbles in this illustration. But once we zoom in on like the key players that make DNA replication possible, we're gonna need to keep the picture simple. Having too many replication bubbles will make everything confusing. So we're gonna actually talk about a replication fork, which is just half of a bubble, okay? So if here's a single replication bubble, okay? Now again, it's easier for me to create animations when things are more linear, okay? So I'm gonna start drawing my bubbles linearly, okay? But it's the same idea. And so with a replication fork, it's just half of the bubble. So this is what you're going to be seeing as we dive into the details, okay? So that's where we're gonna stop this video. The next video, we're gonna talk about the enzymes or proteins involved in creating that new strand of DNA. So stay tuned for that.